There's no. Share screen 2. I'll move this whole panel. Oh. Right. All right, before we get started with the lecture, I just like to give little notes of hints and stuff. I told you last week, I showed you the, I'm just gonna open this up so I can remember what it's called. But right here, you can see due on February 18th. Fit Days, that's the app I had you download last week. That's the name of the app. If you open this up, Smart Body Composition Scale, that was the scale I had. It's now up in the library so that you just go to the library, you can go any day, anytime. It takes about 5 minutes to do the, total from getting to the library, getting the device, getting it open up and doing it. It's like a total 5-minute thing. It's only about 30 seconds to actually do the measuring. So you need to have it done before class. So I have it due on the 18th, which is a Wednesday. The 19th is when we're going to use the data. So that should give everybody plenty of time to get a time to go up to the library. Again, 1 or 2 could do it on a break during the day before or after class or any other time you're up there. If you didn't get the QR code last week, the QR code's also in the box. Like if you open it up, there's a sheet of paper in there that's like, here's the QR code for the app. Do this, do this, do this. It's very user-friendly. There's also a PDF on Canvas in case you need more. So that's what that is. And you find it, the assignment on modules is tucked down in the day that it's due. So it's not till you come down to the 18th. So just kind of a heads up about that. We got a few weeks though, till that happens. But all right, so today we're gonna get going. It's a fun ride today. And talking about theories of aging. Now we're going to be doing theories of aging this week and next week. Chapter 3 is divided. If you're doing the extra credit and you did this week, you can still turn in, because it's divided out, you can still turn in more next week. You don't get 10 points of extra credit. It's still 5 for the chapter. But since the chapter is divided, if you didn't do it for extra credit for this week, you can still do it for next week. Just turn it into the same spot. So that I can still give the extra credit for those who wants to do Cornell notes on the chapter itself. After this class, and same with your study guide will be able to count as notes for exam number 1. But you need to make sure they'll be uploaded in the study guide. So if you add anything today or whatever, either make sure it's in your notes section for Cornell notes, or it's in the theory study guide. Again, only things that are up in Canvas can be used for the exam. So what we're going to start with, because there's a little bit of confusion sometimes. If you're in my classes, I do this like every semester. So you've heard this little lecture I'm going to do for about the next 5 minutes. Hopefully enough times that you can correct people at the party over the weekend. Right? That you've got this down enough that you can correct them. So the first is, what is the difference between a theory and a hypothesis? So I have them both up here. So a theory is a well-substantiated, which means lots of research has been done to explain some aspect of the natural world. It's based on a body of evidence. So it's not an idea that somebody comes up with because they think it could work. That doesn't count as a theory. You look over at hypothesis. A hypothesis is a tentative, testable, and falsifiable statement. Most times when people say, I have a theory, they're lying. They don't have evidence backing up what they're about ready to say. Most people really mean, I have a hypothesis. We've just decided theory sounds cooler, but it messes you up sometimes in science classes because the world uses the word theory to mean hypothesis. Then we're talking about theories and you think it's the theory, like your friend says, I have a theory about their relationship. And that's not a theory. That's a hypothesis about their relationship that you're taking from inside of your head and what you're thinking. Does that make sense? Kind of that difference there? So a theory's purpose is to explain why and how phenomenon occur. We see something happening and we want to explain how or why that happens. It provides a framework that we then can predict off of. We have a theory, we understand the theory that explains how something happens. Then when we have a relationship based on this theory, we expect this to happen. The theory actually explains. A theory isn't a guess. A hypothesis is your guess or your prediction about what's going to happen. So again, hypothesis provides a starting point for investigation. They're used to make predictions. And when you're talking about your friend's relationship and you're like, ooh, I have this hypothesis about their relationship, you're then watching to see if it's right. Everybody comes back and say, I told you they were going to break up in 6 months. That was a hypothesis. That was confirmed. That was not a theory. Next, we have, so these characteristics are what I kind of just said, but kind of bullets pointed at. So a theory offers a comprehensive explanation for what's going on. It's well supported. Lots of research behind it before we're allowed to call it a theory. It's predictive. It does actually work, right? When we use this theory, what we expect to happen happens. And it's progressive, which means as new research comes out, we can then adjust the theory to fit what we now know. So examples, we have the theory of evolution, family systems theory, germ theory of disease. So there's these different theories. Again, they're not guesses. It's not like we're still trying to figure out if there's germs out there. But it's germ theory of disease because there was a lot of research done that demonstrated that we have germs in our bodies that lead to us getting diseases. And that theory can be applied to people who are sick, and it works the way we expect it to. Hypothesis is tentative. It's an idea that's not yet been confirmed. It is testable. You have to have it stated in such a way so that you can see if it's true. It is also predictive. That's where they often get confused. It's predictive. And it's generally specific. Like you couldn't even have a theory about a friend's relationship. Like that's way too specific. Theories are about how relationships between couples work in general, right? Like, and we have a lot of research behind relationships and how that happens. So kind of a common one is increasing the amount of sunlight will accelerate plant growth. That might be something you're like, I think if I give it more sunlight, it will. Or this specific drug, this new drug that we're all inventing in this classroom will reduce the symptoms of the disease. That means if we give the people a drug, we expect it to happen. It's not a theory, it's a hypothesis. Now, the other confusion that people have is not just theory and hypothesis, but theory and law. So I have not changed any words in the theory category. They're all the same ones from here. It just kind of shifted some reason on my slides, maybe to make room for law. Theory still means the same thing it meant. It's still explaining why and how phenomenon occur. A law is also well established, but it's a statement that describes the phenomenon. So this is the one that says why and how, and law is just what. It's just saying what happens. A theory cannot turn into a law. They're not progressive. A law isn't stronger once you get enough knowledge about it. A law is just a completely different thing we're talking about. A law in science is when we're describing what happened. And then the theory is the why and how it happened. So the law says that it describes what happens without explaining why. Laws summarize patterns found in nature and in the universe. They just summarize it. It's just boring facts. That's it. It's not how, it's not why, it's just what's going on. Again, it's whereas a theory is explanatory, a law is descriptive. It states a consistent, universal observation. And like the previous two, it is also predictive. Again, why the three are probably hanging out over the weekend together and confusing everybody. But it's predicting the outcomes of conditions. It's not predicting why it happens. Just predicting that it's stable. It remains unchanged because it's just describing the way the world works. If it technically gets updated, it's because we messed up, not because anything's changed. It's generally very straightforward relationships and often mathematical. That you can write an equation to describe it because it's got to be that predictive that we can just shove out an equation and there you go. So some examples: Newton's laws of motion. An object in motion stays in motion. Unless, right, like everybody knows these laws. The law of conservation of mass. It just works that way. Why it works that way, now there's theories that explain why the conservation of mass works the way it does. And Boyle's law. So we have laws mostly in your math and physics classes. Are you talking about laws? Because they're just equations of saying what's going on in the world and we can describe it that way. In psych and soc, you're nearly only HDFS, only in theories. Because the whole purpose is to describe why people think the way they do, why things happen to children, why relationships break up, why, like, they're really only curious about the why. So you're very rarely dealing with laws. In this class, Biology of Aging, we get to do both. There are some laws that affect things, but we're going to mostly be dealing in theories because while we know bodies age, we want to know why and how. That's really the purpose of this class, is why and how does your body age from birth to death? How does that happen? So that's why we're going to spend 2 weeks talking about theories of aging and theories that affect different parts of aging, because that's really what's behind the why and how. And then after we describe the theories of aging over the next 2 weeks, we'll pause and have an exam. Then, so that you don't have to remember theories and other things, we'll have the theory exam essentially. We'll pause and have an exam, and then we're going to go through the systems of the body, kind of repeating what you learn here, but one system at a time. So that we know how heart is different than skin. But for the next 2 weeks, we're just going to try to teach aging as a big, how the body ages more in general. How and why. Questions about hypothesis, theory, or law to feel more clear, hopefully, than it has in your past? When you were in 7th grade and they just threw all the words at you. All right. And these 3 words may also appear on the first exam. They're very valid since we're doing theories to know what a theory really is and what it's not. So make sure if you need this PowerPoint or a version of this PowerPoint, I've changed it a lot in the last 24 hours. Not a lot, but I've made minor tweaks to it in the last 24 hours. Adding animations and stuff like that, but not content. So there is a version of this I posted probably yesterday. if you need to pull it down and get some notes off of it to add, but you can't add this. Like you have to write out the notes yourself. You cannot upload my PowerPoint to your notes on Canvas and call that your notes. That's not going to work. And don't just copy and paste, because you learn by writing. All right, so I need to do a little review before we get into the theories. Because I'm guessing most of you don't walk around talking bio developments lingo all day long. Neither do I. So to keep us both on the same page, you have two different kinds of cells in your body. And again, the goal of this course is to simplify as much as I can. So yes, I'm skipping some technical details. I'll answer questions, but I'm trying to keep it as simple as possible. But it is true, we only have two different types of cells. We have germ cells, and you either have one or the other. You either have sperm or you have eggs. Everybody's good so far? Nobody's confused. Good. Those are your germ cells. All of the rest of the cells in your body are called somatic cells. Skeletal muscles, blood, stem cells, fat cells, neuron cells, organ tissue cells, everything else is called a somatic cell. And I'm going to use the word somatic frequently this semester because we're mostly interested in the aging of our somatic cells. So make sure you understand what somatic is. That's all of the other cells that have nothing to do with your reproduction, right? We basically, some people call them reproductive cells, but they're technically germ cells, which matters since the book will use the biological terms. All right. Our next review is DNA. Again, you do not need to know the terms in these little charts. They're just the best pictures I had to talk you through. Okay, so I'm going to try talking you through how this works in case you've forgotten. Now, you know, technically it was probably been covered in third grade, but nobody remembers back that far. So depending on when you last had bio, when our cells divide, like we go through cell division, they have to divide at some point. There's phase one. We're not going to deal with that right now. It's just called G1. Then we have the S phase after G1. And in the S phase is when, as you may remember, that we have our chromosomes that are kind of like the double helix chromosomes. They have to peel apart. So they peel apart. That's what this is showing, the peeling of them apart. And it comes in and pushes them around, peels them apart, like the DNA. Does anybody remember what it's called that gets placed on it? Like to make the copy? You have some RNA that helps. It comes, it attaches so that the RNA, and then over on this side, it attaches so that it goes down, it splits apart. And then once it's split apart, it's then going to come over here. Oh, then we have G2 phase. We're not going to talk about what happens there. And then we have the N phase, where those are the, up there, the little squiggly lines are turning into the new chromosomes and the new cells. And so by the time you go through the process, you've now got two cells. But this chromosome, the DNA has to split up and get replicated. That really matters. I'm going to talk about that in like 2 more slides again. And I'll bring up this little picture again so you can remember. Again, you don't necessarily need to remember all of the terms on here. You're welcome to. They'll come up in textbooks. How cool DNA polymerase is. But what you need to know for the theory purposes is that it splits up. You get new copies of each side, and they make new cells. They will divide up. All right. Now, this process of cell division is not uniform. So there is what's called a G. So there was no G0 on this because G0 is when it's not doing this. It's like ground zero. It's doing nothing. So when we're in G0, which means the cell is not replicating, not splitting, just doing its thing as a skin cell, doing its thing as a heart cell. It's in G0. But there are different lengths of times cells stay in G0. And ones that are what's called fully meiotic are the ones that replicate throughout your whole life. Again, at this point, you don't need to know a lens cell from an intestinal cell. Like that's not quite important at this point. It's showing that you have cells that continue to duplicate, replicate, split, go through that process throughout your entire life. The semi-meiotic cells do that, but less often. They're not as keen on duplication. And the last one, post-meiotic cells, after full stage adulthood, and depends on the cell when we consider full stage adulthood, but once we're in full adulthood, they stop cell replication. They're all done. They don't die, they just don't keep doing that process anymore. What is that again? What? Can you explain again? So post-meiotic cells, those are ones that they've stopped doing the cell division. They've done all of that work that they needed to do by the time you reach full adulthood. So your heart cells, skeletal muscle cells, and brain cells, once you've reached full adulthood, they don't keep doing that. But your brain cells in child, that's one reason, as a quick example, when somebody has a brain injury, brain trauma, concussion, as a child, it has very little, depending on what happens, but it has a lot less negative effects on that person. The brain can duplicate, replicate, fix, and change a lot of things, move to a different part of the brain. It can do a whole lot of stuff when they're still children. Once we reach adulthood, the brain can't do that anymore. It can still heal, and we'll learn about what that heals, but it can't do what it could do when it was able to duplicate, replicate, and repair, and do all that things that it was doing when it was a kid. These cells on the far right do that forever in your body. It doesn't age out in that way. And the middle ones are kind of in between the two. They do, but they're not quite as prolific as their cells over here. So that's going to matter in aging. Does that make sense? How this is going to matter when we have some cells that are able to duplicate, replicate, pull apart forever in your life, and others that stop doing that. So how we pay attention to what's going on matters a little bit for us. All right, so altered cell communication. See if I can get my little video clips to work here. This is picking up where we left off. I showed a beginning of a video clip 2 weeks ago, and this is going to pick off talking. So it's only about a minute long. It's a really short clip about one way in which we're going to start into the aging of the cells.
From the moment of conception, our cells are constantly dividing and changing, and continue to do so throughout life. However, understanding the biology of aging is incredibly difficult. There are numerous factors involved in this complex process that are all interrelated. Aging is an inevitable time-dependent decline in physiological integrity and function of various organ systems, caused by the accumulation of cellular damage.
So we're going to jump in. So I just wanted to start it there. And then what we're talking about specifically.
Cellular senescence, mitochondrial dysfunction, loss of proteostasis, telomere attrition, epigenetic alterations, and finally genomic instability, which is one of the major players leading to cancer. Let's first take a look at altered cell communication. This involves a gradual and harmful change in chemical signals between cells. This signal degradation impacts on how cells behave as individuals, groups, and the surrounding microenvironment. So cell behavior affects the cell environment, and the environment in return affects the cells. As we age, the signaling environment of the chemical messages across the whole body tend to become more chronically inflammatory.
Right, so what it's saying is these cells in here, they're going to need to be communicating in the body with each other, right? Things have to communicate. And as things become inflamed, so we have inflammatory processes in the body, it's harder to communicate. If you think of it about being like a fuzzy signal, that it's not communicating, it's just struggling a little bit. So that intercellular communication is getting interference and having a little bit of problem.
Inhibiting the immune system and potentially causing other effects like muscle wasting, bone loss, and impaired neurological function, as well as other harmful effects in a process known as inflammatory aging or inflammaging. Multiple different factors cause inflammaging.
And we're going to come back to this here in a second. There you go, that worked. So one of the processes that's going to play a role in the aging is going to be the altered cell communication. Right, that's more of a key term. We have theories, but we have some key terms that we're going to talk about that are going to help us out. Meaning that as we go through development of the body, as we age, the communication from cell to cell gets fuzzy or there's interference. Everybody's played the telephone game, right? Where you whisper a word into somebody else's. Have you done it when there's lots of noise and other thing going on, like leveled up, so you really have a hard time hearing? It makes it very different, right? There's a much better chance. I've actually also played it in Denmark before I could speak Danish. And so somebody had to translate the word for me into English, then I had to tell somebody else in English, or the sentence, and then they had to translate it. Our team did horrible. It was really kind of funny. And then we decided one time we found all the bilingual people of different languages and split us all up and did like retranslations, you know, from English to Danish to English to Spanish to English to Portuguese to English. Like it was a mess. Now Google Translate can just do it for you as you click through languages. It can mess things up pretty fast. But that's what's kind of going on in your body as you age. So if you get a wrong signal, it might do a wrong thing. Right? It's not what it meant to say. It's not what the body told it to do, but it might be like, oh, I'll go do this. So that's one of the things that's going to affect our aging. Cellular theory, this is one of your theories on there. So if your theory study guide ready, that should also be open, if not in a different spot, so that you're ready to go on this, is we have apoptosis. And this is cellular. So up here, I have a killer lymphocyte. That is floating through the body. If you just pictured that last thing of things floating through the body, we have killer lymphocytes floating through your body, looking for cells that look suspicious. And if it finds a cell, so this is a different cell down here, that looks suspicious, meaning it probably has a virus or something else going on. There's something going on in the cell that is problematic and it doesn't like it. So it comes in and it drops down into these receptors here. And then the receptor is named appropriately. A death receptor or a death-inducing signaling complex is what's going on. It's going to go through the process, and then we're going to come down to apoptosis. So that's from external. So a cell outside comes over, drops down, and says, I'm going to kill you. And if you think of apoptosis, you can imagine it just pops. Right? The cell goes pop, it's all gone, and all of its components get absorbed into the body. doesn't really disappear. It just pops and goes different places. So if you picture video games, I think a lot of video game stuff going around where you're chasing around, you're looking for the right ones, you drop down and you pop it, then you go around looking for another bad one, drop down, pop that one. So that would be extrinsic, external, like something's coming around, finding the bad cells and popping them. They got a little pin and needle, pop, 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 pop. So that is extrinsic apoptosis. Cells can also experience intrinsic or internal apoptosis. As you can see down here, I have on the slides the individuals that are also from your textbook. And so this was actually discovered back in 1842. So we've known about apoptosis for quite a while. So in the intrinsic one is there's actually stress going on to the cell. Rather than there being a problem inside of the cell's just getting super stressed. There's lots of things that can stress a cell. But for right now, we're worried about we have the stressful cell going on. And so the stressful cell recognizes it, picks a non-stressful cell that it's by to help it do the process, grabs the protein over here. It's called BH3 only protein, and it goes to, again, it doesn't matter what the terms are called. It comes down this way into the mitochondria, and it pulls out the cytochrome C out of the mitochondria. So it took out a key component. So if you're badgering this time, the cell inside pulls something in to mix inside, and then it kicks out this cytochrome C, which goes through the process over down here. Like it's pulling it out here, puts it in. If you picture that little green thing in, circles it around, adds in caspase 9, circles it around, and that actually will then inside of the cell make it pop from inside. We still hit apoptosis at the end. The apoptosis part is the same, that it pops and then it gets absorbed into the body. But one are these killer lymphocytes running around popping things, and one is an internal process where it recognizes that it's super stressed and like pulls things around, and then so it can pop itself. If you've ever felt that way when you were super stressed, you just wanted to like blow up, right? Just pop. So that's what your cells are doing when they get super stressed. And then from there, that's from the outside. Questions about how apoptosis works? Okay, I'm going to give you a second, and I want you to tell the person next to you real quick how intrinsic apoptosis works and how extrinsic works, how the two different ones work.
Okay. And then.
Okay, any questions now? I'm happy to take, because if you're still curious, or everybody wants to see if they got the right guess, and you want to try to explain them? Okay.
So for intrinsic, you have a BH3 protein and a BCL2 protein. And if the BCL2 protein is anti-apoptotic, then it becomes inactive. And if it is an effector, if it is active, then apoptosis happens.
Yeah, so yeah, here you're right. Here it gets blocked, and we're doing good. Here we're coming down and pulling the little green things out. And we need the green things out in order for it to pop. Because in apoptosis, we want our cell to pop. If you think about it, again, if I use a balloon as an example, hopefully this will help. If we pretend the little green things is like kind of like changing the air density inside of it, like if you suck it out, it's gonna pop. And then over here, it's more like poking a pin in a balloon. And then it pops. Something comes from the outside. This external killer lymphocyte is popping it. And the other one, it's inside and it's like, ooh, I'm super stressed. Pop. Sucks out the air and it pops. But either way, does this cell duplicate? Is it duplicating? No, it's popping. It's no longer a cell able to duplicate because it just popped. But its components will go back into your body. You will reuse them in fun ways. But yes, this is cells popping. All right, now I have another review before the next theory. Anybody remember the Hayflick limit? The term ring a bell from years past? So the Hayflick limit, so Hayflick and Moorhead, everybody, Hayflick and Moorhead were studying how to turn normal cells into cancer cells. They really want to study cancer, but they thought, what if we could take a cell and turn it into a cancer cell? And they studied human embryonic cells because in 1961 you could do that sort of a thing. Now we have different laws protecting embryonic cells, but back then they could just do whatever they wanted to. You'll learn that in other slides today, that we didn't quite have the ethical balance we have today. So they took these embryonic cells. The reason they picked embryonic cells is because they don't have all the environmental exposures. So they were really trying to control and see what's really going on in the cell. So, I mean, it was a good choice. Well, maybe unethical. It did do what they were looking at. And they took female and male. Now, they're not red and blue, but they look differently. So he could tell them apart. There's nothing significant about them being male or female. That doesn't really matter, other than it meant he could watch the cells and see what happens because male cells look different to him than female embryonic cells. So what they did is they put a mix of male and female embryonic cells and saw that they did that whole cell division. That's the doubling, cell division, cell division, cell division. Look at all this happy cell division going on. They're skipping around, cell dividing, nothing's popping. Everything's just dividing happily along. And then they pulled out the female and male mixes. So they divided them out. And then we see 30 additional doublings. And pulled out the female only at the 10th. So after 10 times, they pulled out all the female, and it did it 30 more times. So this was, and then they pulled out the males, they did it at the 40th, and it did it 0 more times. So this one, 10 plus 30 equals 40, and this one was 40. That's the important part of the story. And what they found was that they just stopped duplicating at some point. Like nothing happened to them, like nothing interfered, nothing came in. This was in a dish, wasn't in the body. So there was no little thing coming over and popping stuff. Didn't pop from the inside, it just stopped duplicating. It was just all done. I'm tired, I'm not going to duplicate anymore. Take that. So that number of how many times it can duplicate before it just stops is called the Hayflick limit. It's the limit on a cell of how many times it can do this duplication. Duplicate, 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 stop. And we can see that different species have different Hayflick limits in their cells. So bats, it's about 16 to 29. Chicken and horse are both there around the 30 to 40. Humans are 45 to 60. Mouse is about 12 to 15, and rabbit is 21 to 27. People thought for a while it's not a theory because it doesn't actually explain why. It was suggested that maybe it could be a theory explaining why we age, and that maybe organisms that had more doublings live longer, because we did see that, because we start with mice. Mice are 12 to 15 and live 4 to 5 years. Humans are 45 to 60 and live 115, 120 years. So clearly, I mean, it was an observable difference, but then it gets all messed up. Chickens are 35 to 40 and only live 6 to 10 years. So that doesn't actually explain aging. A number of times in and of itself in a species doesn't explain, because when we study aging, we look inside of humans, of course. A little bit geocentric, it's probably the species we're most interested in. But to explain it, we sometimes take the idea or the hypothesis, apply it to a different animal, and see if it works. And if it was true that the Hayflick limit itself is what was predicting how long a species lived, then we would have seen a better pattern here. But it still matters. It's still a very important concept that's going to play a role in some theories, is this Hayflick limit. That's the number of times a cell can do the duplication, and then it gets too tired. It stops. I'm going to jump over. If you look at your theories, I actually pulled this one out of order. It is a genetic theory, but it helps the rest made sense, so I moved it. And this is a genetic theory of telomere attrition. And it's because it uses the Hayflick limit. So telomeres, remember I said this slide was going to come up again with the DNA here, unzipping our DNA. Telomeres is the repetitive, non-coding base pair at the end of your things. And if you remember, you have like the TAGG, that all those TAGCA things, that those are your DNA codes. So it's trying to say that you have, like, if we imagine you have that pairing and it comes down, we have telomerase that comes in here. That, do you see how it makes it longer? How this one makes it longer? Can you see that? This is the important part, that the letters inside of this does not matter. And it then keeps the same thing. Do you see that? And so this beginning one is the same as this one, and is the same as that one. Do you get that? This beginning, TAAGGTAA, blah, 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 across, is the exact same as right here. No change. Everybody see that? There's no change from this top germ cell to this bottom germ cell, because of something called telomerase, which is like an extra little, oh, don't forget these letters, oh, don't forget these letters, because our germ cells always have to be perfect. And we need them to be able to, like, keep working the way they need to work. Not perfect in action, but perfect in DNA. So this one will have something called telomerase, which will get it to keep, but you only have a certain number of germ cells in your body, but you need them to be able to do what they do. Somatic cells, that's the rest of your body. That's not either your eggs or your sperm. So this is eggs and sperm cells over here. This is everything else. When it goes through a cell division and splits apart, what happens to the letters? Does it have the exact same number of letters in the next little bubble than the one before it? Nope. And then is the next one got a little bit less? And then we run out. Self-senescence is when it runs out. So these are telomeres that hold these. So it's not, it doesn't mean your whole chromosome runs out, but your telomere. And I got another picture to show you. So unless, here's the important part here, unless the telomere length is maintained by the enzyme telomerase, it will get shortened every time. And it looks like this. you look right here at the picture first. Look at the picture first. I couldn't get it all on one slide without it getting too messy and confusing. This is that getting down. This is this right here. Is this right here. See, it's big purple, less. So it's just the end cap. Most of your chromosome is staying intact. But the little tip, every time it unzips, it doesn't quite unzip all the way and it loses a bit of the top. And then the next time the cell divide happens, it opens up, but not all the way and loses a bit of the top. And that is how the Hayflick limit works. When it runs out of telomeres in the DNA, then it just can't duplicate anymore. It can't unzip properly, so it stops. Do you see how the two work together? So they first discovered the Hayflick limit. They just knew it was a thing that happened, that your cells can duplicate so many times and then stops. And now from the DNA level, we can see how it's working, that the telomeres are shortening. And as the telomeres shorten, it stops duplicating. Again, this is not apoptosis we're talking about. It doesn't pop, it just stops duplicating. So rather than apoptosis, that's programmed cell death, Hayflick and others predict there's a limited number of times. In humans, the number of times somatic cells can replicate is regulated, maybe not entirely, but a lot of it is regulated by this telomere length. So we're going to watch, go back to our little thing and go to 8:41.
We also have a biological clock in our DNA, which has an expiry date. The body is constantly going through cell divisions, and every time cells divide, they make a copy of all their DNA as well. DNA is tightly packed into chromosomes. Due to the imperfect nature of DNA replication, the ends of the DNA are often skipped over. Chromosomes have regions at the ends that are protective caps called telomeres, which contain non-essential information of a specific DNA sequence that is repeated thousands of times. The sequence has two purposes. Firstly, it protects the coding regions of the chromosome, preventing them from being damaged or fusing with other chromosomes, and ensures that no genetic information is lost. Secondly, it acts as a clock that controls the number of replications a cell can make. This region gets shorter every time replication occurs. Because this region has a defined length, the cell can no longer divide after this point. This is known as the replication limit. The replication limit of most cells in humans is roughly 50 times. This limit also helps to prevent cancer, which is the opposite problem of uncontrolled replication. When telomeres reach a critically short length, cells sense it and permanently turn off their replication machinery and senesce. An enzyme known as telomerase, which is turned off in most adult cells, can prevent telomere shortening and even restore telomere length. The presence of telomerase is one of the hallmarks of cancer. Thus, telomere attrition limits the number of times our cell can divide, slowly leading to dwindling populations of functional cells. If you think about it, every cell...
Okay. Okay, so then back to that work? Nope. I guess I need to do this again. There we go. Now it took me back to the beginning. Mine will take us back where we were. We're right here. So this is the telomere attrition theory of explaining aging. So that's in your genetic theory section. But I moved it up here just because it kind of made sense when talking about cell replication. So go ahead and explain to somebody the concept of the Hayflick limit, and then how the telomeres work, like how these things, the replication or Hayflick limit to somebody next to you. So you can explain this before we go on.
And then it covers all good, like, certain, it's about. Yeah, they break it like. Yeah. Just basically. She said, about this right here, that was there.
Yeah.
All right, let's have somebody try to explain it. What do you think? I'll pick. All right, this table over here. Go ahead and try. You can be wrong. You'll learn it better. You'll get better grade on the exam if you guess wrong than if you don't guess. Okay. Go ahead.
The Hayflick limit is the set number of times the cells that duplicates before it eventually stops.
Mhm.
And the telomere theory states that when the telomere loses its length, eventually after each division, that's when it reaches the Hayflick limit. And when it stops,
Yeah, then the cell senesces, which I'm going to cover next. So you're correct. That is right. So that the Hayflick limit just says the cell only duplicates so many times and then stops to about 50 in humans. And what plays a role and how fast we reach that 50, right? Like, is how fast these telomeres go down. So for example, why do some people get gray hair earlier? And some people get it longer. It's the same 50 replications, but the length of time it takes the telomere to go down and the time it so that it replicates again. What pushes a cell into needing to replicate, takes it out of G0 and throws it into having to do replication. We're going to talk about what causes those. And then if you speed up the process, so when we go through the different systems of the body, I'm going to talk about things that affect telomere shortening. Why your hair cells or why your skin cells or why your heart cells, why some of those age at the rate that they do is because there's things in the environment that affect our telomere length. One general example, because I know we have a lot of psych and HGFS people in here, a lot of research out there shows that childhood trauma affects the telomere length of children. So when they go into adulthood, they have shorter telomeres than others. Now, there's things you can do in adulthood to slow it backwards and speed it up. So it doesn't mean those who had childhood trauma are always aging faster. Does that make sense? It just means if you had a lot, and childhood trauma and includes things like death of a parent, an incarcerated parent, like their child abuse, like their severe childhood traumas are what we've studied, the severe childhood traumas, affect the telomere length. So that's just an example. We'll go through the body systems and look at more examples. So what happens since it doesn't pop, there's no apoptosis going on. Instead, it's called cellular senescence. And this is a key term. It's not a theory in and of itself, but it's a state that it can go into. That, as I show here, is not apoptosis. I put that on there so you know that's one route, but we're talking about how it instead goes to senescence. So this is damage to the cells in some way, whether it reaches the Hayflick limit, it can go into senescence. Things happen. Senescent cells are actually resistant to apoptosis. They can't pop. So a normal cell can have something happen to it, either come along, get sick, and it can be popped. It can be stressed, it'll pop itself. Or if instead it senesces, then it can't. It's apoptosis protected, which means it doesn't go back into our body stream. It just hangs there. Your senescent cell is just kind of hanging out, looking kind of funky in your body. So a really healthy immune system can target senescent cells and kind of remove them. It doesn't pop them, but can remove them and kind of recycle them and do some things with them. But you gotta have a good immune system for that to work. And you gotta have good communication to tell it what to do. So we talked about the communication, right? We have some cellular communication. When you have a lot of senescent cells, it's harder for the cells to communicate, and it's harder for the cells to communicate. It's harder for them to know to break down the senescent cells, to sell things, send after it, to clean it up. So when the body can't keep up, they begin to accumulate. And we're gonna see what happens here. I'm gonna go back a little bit to where we left off.
So cell behavior affects the cell environment, and the environment in return affects the cells. As we age, the signaling environment of the chemical messages across the whole body tend to become more chronically inflammatory, inhibiting the immune system and potentially causing other effects like muscle wasting, bone loss, and impaired neurological function, as well as other harmful effects in a process known as inflammatory aging or inflammaging. Multiple different factors cause inflammaging, one of which is the senescence-associated secretive phenotype, SASP, which is directly caused by another hallmark of aging, cellular senescence. As cells replicate, they eventually enter a phase of permanent non-division and cell cycle arrest, when they run out of replicatable DNA at the chromosome ends. However, it can also occur as a result of damaged chromosomes. Senescent cells normally destroy themselves via a programmed cell death mechanism called apoptosis, and are removed by the immune system. But the immune system weakens with age due to chronic inflammation and immunosuppressive environments. Increasing numbers of senescent cells escape this process and begin to accumulate in all tissues of the body. These senescent cells are known to secrete inflammatory, immunosuppressive, and a harmful mixture of factors, SASPs, that have been shown to encourage neighboring cells to become senescent and may contribute to multiple age-related diseases. The leakage of...
So what they said here that was really important is that when you start getting the senescent cells in your body because you reach that 50 Hayflick limit or maybe other damage, we'll talk about how else, there's other slides that will end with senescence. But right now we're doing the Hayflick limit reason that you reach cell senescence, but they're contagious. If you don't get rid of them soon enough, they're like, hey, you should be senescent like me. Even if you haven't reached your Hayflick limit yet, you should senesce too. But if your immune system can keep up with it and take care of it with like your, we'll learn about it later, but the T killer cells, right? We have some things that go in and clean up. If we can keep cleaned up, we're good. And if we can't keep cleaned up, then they're gonna start. Again, if you picture a video game, have you ever been at the one where Tetris or anything else, once it starts coming, it just starts coming, right? It starts speeding up and then everything's good. First, it's nice and slow. You have early stages of just about any video game you play and you're good. And then you get better and then it goes faster and faster and faster and faster. That's exactly how senescent cells work in our body. At first, a couple go, we can clean it up. This is not a problem. And then they start coming so fast that our body can't keep up with it. And that's leads to the aging of the body. I just click over here, that's right. Yeah. So, questions about senescence? Again, senescence is just the cell stop. Fire?
Is Hayflick more at the nature proponent for telomere attrition.
Correct. And again, because that's what we discovered in 1961, is that cell senesce, right? Like, so that's who did it. Hayflick figured out that that's kind of who got us started on it. So that one we haven't known quite as long. 1961 may seem like a long time ago to you. But to most of your grandparents, it wasn't that long ago, at least. Or yeah. So, as far as research goes, a lot of older adults today don't even know this is a thing. They would not have even had it available to learn in biology if they wanted to. And they take, like if they had taken a class, it wouldn't have been there yet to learn about it. So as was hinted at in the little video clips I've been showing, senescence and cancer have a love-hate relationship with each other. So you may have thought, I'm actually going to go back to here, that telomerase is what saves the cell. So many people think, okay, so if we know we can add telomerase to the tip and it will grow the telomere back, why aren't we painting on telomerase onto all of our telomeres, right? Like, doesn't that seem like a really good idea so that we could not get gray hair or wrinkles or any of those other problems? We could just be painting on telomerase all day long. It's an enzyme. Pop an enzyme pill, take it in there, make that work better in our body, and off we go. Unfortunately, some of you already have cells that do that. Because if you have cells that do that, it's called cancer. Cancer means the cells pass the Hayflick limit and just keep on trucking. They don't senesce. They just duplicate and duplicate and duplicate and duplicate and duplicate. Then you get a tumor. It's a slightly overexcited cell. It's got like, hyperactivity on the high-end scale. So cell line in vitro cells that can duplicate forever. Now I got to tell you a little bit of backstory probably because I'm also an HD vascular, but because I think Henrietta needs her time in the sunlight here. So George Gay was looking at malignant cervical cancer tumor cells from Henrietta Lacks. We now call them the HeLa cells. In 1951, did we have a lot of medical ethics back then? No. So he's like, I'm just going to take those. So he took herself. Out of her body, put them in a petri dish. Simplest way to explain it. And they continue duplicating into its own tumor outside of her body. While she also died in 1951 because he got the cells, and that's really what the doctor was most interested in, her cells continue to duplicate in vitro and are actually the cells that gave us cell biology. They are still in use today. Her cells are still duplicating. And they're called the HeLa cells after Henrietta Lacks, but George Gay gets all the credit for it. Most of the time you just see HeLa cells and you don't know this poor woman who died of cervical cancer, whose body's given us just about everything we're going to learn in this class. It was her cells. Has her family received anything for those cells? No. No, just his family. He gets all the credit and all of the things. So, a little bit of my own history moment, so we remember to give credit where it's due. So essentially, short term, anybody who's into the gods, whether it be Christian gods or Viking gods, I don't care which gods, but if we're talking immortality here, the idea of eternal cells that can replicate forever totally fits within science. Within a mortal body, doesn't work. That's cancer. So people play around with sci-fi. Could you play around with sci-fi? Cancer is kind of the immortal cell, but somehow it doesn't work with our body. But it is a cell that duplicates forever, and doesn't stop. Again, her cells are still trucking. And she died in 1951.
How are they keeping it alive?
They just keep doing it. They don't keep it alive.
They just,
The cells just keep duplicating.
where do they like holding it at?
In research centers.
Like all of our houses only one, is there only one place or?
No. People buy them to do research with.
Oh.
Yeah. Yeah, and that's why they're called HeLa cells. Like, I can read research on HeLa cells. And what they can do with that.
Won't the cells eventually run out of energy?
Well, but they keep duplicating to new ones. So we still have more.
New ones still with cancer.
Well, because nothing has cancer. Cancer is just a cell that keeps duplicating. It's more of a description than a thing. You can't get cancer. We say that, but you can't. Cancer is when your cells just keep replicating and replicating and replicating and replicating and hit the 50 and keep going to 100 and to 150 and to 200 replications. And your body, its processes have meant for it to stop. It's one reason why older individuals get cancer more often. Because you have to, whereas younger children with childhood cancer looks differently because it starts sooner, like that it went really fast. It's like a really fast aging of the cell. And then it's now replicating, right? So childhood cancer is a lot less common, but more deadly because of how it's approached. Whereas older age cancer is pretty much just that you hit the Hayflick limit at the normal age of everybody else, but then your cell kept going. Either it produced telomerase that it wasn't supposed to. We're going to talk about different reasons why then cancer flips in. What's your body? Why is your body so cool that it wants to live forever? So anybody who's got cancer, their body's just really trying really hard to live forever. It's just that cell is like, I could be eternal. I could do it. I am an immortal cell. Stuck in a mortal body. It's just not gonna work. It kills the mortal body to have an immortal cell. So senescence is not like the predictor of aging. That's why it's not a theory, because it's more of a key term, because it's a mechanism that actually suppresses tumor growth. We need our cells to senesce, or we create tumors in our bodies. So as it says here, cancer cells, and I have this all in here because some people like to go find this again because they're trying to tell their parents about what I just said, and then it doesn't make sense when they say it. So they need to come find my PowerPoint. So that's why it's a boring slide, because I wanted the details in there for you. Cancer cells often avoid senescence or cell death by maintaining their telomeres despite the repeated divisions. It's possible because the cancer cells figure out how to activate. Because all cells are the same-ish. We have our germ cells and our somatic cells. And technically, they're supposed to know not to use the telomerase in the somatic cells, that only the germ cells should use it. But somehow the cancer cell turns it on, turns on that telomerase. It's like, ha ha, I figured it out. So it adds genetic units onto the telomeres, makes them longer again, and prevents them from shortening. And I remember when I first learned this, I thought it was really kind of crazy that I always thought cancer was like a bad thing killing people, but really it's trying to live forever. And that really messed with my mind, like in a fun way. Like kind of a...
What does chemo do? Does it kill the cells?
Yeah, it damages them so they go into senescence. And when we learn more about what some of these other theories will help make that more sense, where chemo comes in and is like, tries to push it either into apoptosis or senescence, like it's shoving that cell into that state to try to make it so it stops replicating.
What if scientists, I mean, I don't know if it's probably did, but scientists try to put the immortal cells into different species, it's not human. Would you think they would still live forever, or no, it wouldn't work like that?
Well, I don't know of any research on it, but that's not my area of expertise. But that aside, from what I do know is we do sometimes put organisms in different ways. There are things that we do. But even when you have an organ transplant, human to human, most bodies will fight the transplant. Like you have to take medication so your body will, and we don't even know if it will accept the kidney or accept, right? We talk about that. So in theory, it would fight it. Like just any foreign thing brought in, you have a heart transplant, the body will try to fight that new heart at first. You've got to try to train the body to teach the body that that heart is okay. Because it knows it's not the original heart that goes with the body. But medicine has done a lot of things. So it would try to fight it, that I do know, but there's not as much interspecies. Often we just duplicate it in the species. So we have mice that also have cancer and we have their cells. Like we have all sorts of, the HeLa cells are just those ones. But there's tons of cancer cells that we use for research and stuff too, that continue to duplicate and do other things that we can use from mice and other animals. And those are more ethical to use and do things with. So. Okay, any other questions about senescence and cancer?
So you said that like, I think that was something else. But the ones that their telomeres are going down, like the ones that telomeres are shrinking,
Yeah.
that they're kind of like contagious.
When they senesce.
Yes, would that be the same for cancer then? Or no?
Or do they send a signal telling the other cell? Generally, I don't know for sure, but I don't think so. They don't make other cells, because normally when they say the cancer cell is spreading, it's the one duplicating. And when cancer spreads, it's whatever's going on in your body. So it kind of does and kind of doesn't. It's part of that cellular communication that it's talking to the other cells. And if your body knows how to turn on cancer, like it knows how to turn on that telomerase and use it, then it may be able to do it in other parts. And that's why they try to get rid of it. Because that cell can get out and start duplicating other places. In most cases, the cancer is the same thing. It just has moved to another part of your body and made a tumor over here. But just like her cells are still duplicating, it's the same ones duplicating. That's why they try so hard to get rid of all the cancer. Because then that cell is no longer in your body to be able to duplicate somewhere else. But if they don't get it all, it'll just start replicating again. And it may be good for a while because it has to replicate before it gets to the tumor level. Does that make sense? So that's why people go into remission. And sometimes we got rid of the cancer, we're okay, but that's why you have to go to checks all the time. Because it's really hard to tell until it had a chance to duplicate up again, if it's going to keep going. And where that cell is going to replicate and affect other cells. So they kind of can in that contagious way, but it's more in the communication. Yeah, but it's mostly that same cell duplicating that causes the problem. So that is so this is all underneath kind of like telomere attrition. Senescence is a concept, so we'll come back to it again, but it came up there. So the next theory we're going to talk about is oxidation. Now this one is generally more interesting to my people who work out or those interested in health. How many of you heard the term antioxidant? What's something high in antioxidant?
Blueberries.
Blueberries?
Grapes.
What's some other good antioxidants out there? Any dark chocolate lovers? Dark chocolate's good in antioxidant. They have those shakes you take that are high antioxidant shakes. Acai from Brazil, that's got good antioxidants in it. So we're going to talk about antioxidants and oxidation at the cellular level. Why in the world are they trying to tell you to take antioxidants? So that's what you're going to actually learn today, right now. So oxidation, it's actually one of the most popular theories because all of you have heard about it. The only reason why there's commercials about antioxidants is because of this. So you technically, if you buy antioxidants and think it's going to help your body, you believe in the oxidation theory of aging. You subscribe to that even if you didn't know it had a name. The 1956 that the theory was named. So we're going to play. So if we look up here at my tomato, because you may have seen a tomato be oxidized before. When you have a tomato and then it gets kind of super ripe and starts turning brown, that's the oxidative process starting in the tomato. And then it turns, if you didn't take care of it, it kind of shrinks up and turns into this like gross looking thing. That's oxidative stress on your tomato. Inside of a cell, we see kind of the same process. A cell can get attacked by free radicals. I'm going to teach you how that works. And then it looks like a messy cell when there's a bunch of oxidative stress, right? That is not a happy cell. So what happens? We now get to jump over to your chemistry years, from your biology years to your chemistry years. So you may or may not remember that electrons like buddies. They don't like to be alone. Do you remember that? We talked about having electrons on things. We have paired and unpaired electrons, and you have like the negative if it doesn't, all that kind of stuff running around. What really matters is that when we have a substance becomes oxidized when it loses an electron. Now, I said my workout students like today, this theory, because one of the things you talk about, how do you build muscle? What do you have to do to the muscle?
There.
And you have to oxidize. That's how it tears. You have to get that muscle to lose electrons and go through oxidative stress in order to build it. Now, random note, we will come back to this when I teach the muscle section. This won't be the first time, it won't be the last time you hear it. But if you take antioxidants before working out, you can't build muscle. Because you just took antioxidants and you have to oxidate. And so your body has a whole bunch of antioxidants ready. Because what an antioxidant does is it throws electrons at your oxidated substance.
When you say build muscle, is that just the size, or is it also how strong it is?
Also how strong it is. Because when you're working out and trying to build muscle, both the size and the strength of the muscle, it tears. But part of that, what we call tearing, is really oxidation. So you don't want to do it before you exercise. You want there to be the time. You don't want to take those shakes beforehand. 2 hours later is great. Rebuild your body because you don't want things to stay in an oxidated state. You want to neutralize them because things that have, one electron missing is not so hard, but if you're missing more electrons, that's when it gets called a free radical. Is when it's oxidized and lost lots of electrons. So it needs more to keep it. So when you hear about the antioxidants, really what they're trying to do is we have free radicals out in our body, and we try to throw antioxidants at them so they don't attack the cell. We're trying to we don't want them to be oxidated for too long. A little bit of oxidation is good. Stress is good for our body. We need a little bit of oxidative stress, but too much is bad.
Is that what electrolytes are?
Electrolytes are not antioxidants. They work in a different way, but they are also helping the cells through the process. It's a cell with a different problem. So here, we have free radicals or our reactive oxidative species. So that's where ROS comes from when we see it in the book. Are molecules that are generated during the oxidation process. And again, oxidation needs to happen. We need our body to oxidate, reduce. Reduce is when you get one back again. Oxidate, reduce, oxidate, reduce. We need that. But they have the unpaired electron, which makes them unstable and extremely chemically reactive. The more electrons they're missing, the more crazy they are. More severe oxidative stress can cause apoptosis. That's internal, intrinsic apoptosis. Because it gets looking too crazy. Remember when I said when the cell gets really stressed, it intrinsically pops. So then you need the antioxidants in your body to stop it. You know, gets here, we need antioxidants here so it doesn't get to there. We need it to get a little bit so that it starts building, growing, and doing. Our body needs to go through that process, but then we need it to stop. All right, so go ahead. Oh, I got one more. I thought I had another slide on it. Not click it. Let's get it clicked back over here. There we go. So there are 4 lines of evidence that support this theory. Longer lived species mitochondria, and if you kind of remember when we talked about those green things coming out, they were coming out of the mitochondria. So they produce fewer free radicals. So that would suggest that if you have fewer free radicals pushing your cells into apoptosis, then you could live longer, and we do see that at species level. So species, unlike the Hayflick limit. Increased levels of lipofuscin, which is like a cellular waste product, increases with age with this. There's correlation between oxidation levels and deletions in the DNA. Like, so we see that it also affects some of the telomeres and other things. When you have high oxidation, it can also kind of eat at that telomere to make it shorter. And muscles with sarcopenia, which we'll cover when we get to the muscle unit, or atrophy. We see that they have mitochondria with lots of damage in them. So we see evidence of this in the body. And I'll show you the review before we tell each other about it. That was 6. Starts at 6
with age. The mitochondria are organelles within cells that are known to be the powerhouse and the main energy source for cellular processes. They have their own genome, which is prone to damage because it is stored in an oxidation-prone location, and they do not possess their own protective nucleus. Thus, they are exposed to all of the elements. Unfortunately, mitochondria also produce the most free radicals, such as reactive oxygen species, as a byproduct of normal cellular metabolism and aerobic respiration. This results in the progressive dysfunction of their cellular processes over time, where they release increasing amounts of free radicals, leading to inflammation, stress, and cellular damage. These free radicals damage all molecules they encounter, from proteins to DNA, causing them to mutate and thereby dysregulating their function. Dysfunctional mitochondria produce less ATP, reducing the energy supply needed for cellular processes. They are also unable to replace themselves as quickly in their dysfunctional state. The reduced numbers of mitochondria and failure to dispose of defective mitochondria further lead to cellular damage and eventually aging. Proteins regulate virtually all cellular reactions and processes and provide cell structure. The maintenance of all proteins in their original form, folded in precise...
Okay, we're going to come back to this in the next one. So go ahead. Here we go. And explain oxidation and antioxidants in the cells at your table. And then we'll let that table over here, you guys can explain it.
That's why I was, you also said, no, I know that parts of it.
There we go. I also feel that it's not good to go to the body for the north faster. Like, I'm wondering if that.
One, stop it, you stop, you want to stop cities. So I think.
Probably the plus bar, this one means pounds and those reasons. So then we have this.
I think that my friend.
Oh, that's the trouble.
Any questions? Questions?
What is the actual process of oxidation?
So the process of oxidation is when you lose the electron from the
My question
molecule.
is
Why?
yeah,
Why do you lose an electron from a
does
molecule?
it create power in the mitochondria?
Well, in the cell, yes, the cell needs you to have, the mitochondria needs it to lose to do its process. It's part of, it actually originates. Anybody remember how photosynthesis works? Can we go back to photosynthesis? Can you pretend you know what photosynthesis means? Right, that's the plant where it's taking the sunlight, and then the sunlight, photosynthesis is it taking the sunlight and turning it into the nutrients. Oxidation in our cells is this, came from photosynthesis originally, like evolutionarily, it was photosynthesis first. And we don't do photosynthesis, but our mitochondria, what it does is the oxidative process. So it's when the things come in and it then converts it into the energy and the things that it needs, just like photosynthesis works. But if you understand the idea of photosynthesis very generally, you can picture that's what's happening inside of your mitochondria. You need that again, not from the sun, but what's going on internally.
And oxidase, it like stops the process and then.
Well, if it comes too soon, like if as soon as it throws an electron, another one comes in, it didn't get to do it. Right, you don't want it to fill up. Or for the antioxidants to be throwing electrons out there, and then everything always has too many electrons. It never can get rid of it to get down to what it needs to get down to. If you have too much antioxidants in your body, then your body can't oxidate it the same way. So you don't want to always have high levels of antioxidants in your body. And in short, it's like moderation in all things. It's one of my favorite answers in this class. Too much of anything usually is bad. And it's true with antioxidants because we need the oxidative process. But we don't not want antioxidants because we need those too. I'm only talking about like the building muscle part. That's the only one where you work out, then drink the shake. Don't drink the shake, then do the workout. Right? Like, so that you're getting the antioxidants after you do the workout, not beforehand. If we're talking about antioxidant shakes, right? We're talking about the antioxidant things. So yeah, go ahead. You want to explain it? Your table?
So, how oxidation is a normal cell and the free radicals come and like steal from it. And then the antioxidant like donates towards it and then,
It calms it down.
yeah, and like recovers it.
Yeah. Yeah, picture a hyperactive child tearing down all of the things. Then you just calm them down and they stop tearing down all the block, right? Like you calm them down a little bit. So the antioxidants help calm down. But if we can't calm it down, then it will go into apoptosis. It gets to here, that's when it's the too stressed cell. This is the too stressed cell that we saw in the apoptosis slide. Instead of being able to have the antioxidant comes in and going back to being a normal cell.
Happened in like 10 seconds.
Depends on the cell. Yeah, but yes, it can go quickly. Depending on the cell. But it depends on what stage you're talking about too, because the antioxidants have to process through your body, right, to go into the bloodstream and everything. So it depends on where the cell is we're talking about. Because again, today we're not talking too much. I'm using examples, but we're really talking generally not about body systems. So when we talk about the muscle system, muscular system, we'll talk about how those cells and how long it takes those cells for things to happen. Okay. And right on the tail of antioxidant is the theory of nutrient sensing. At least I think these sound a little more like you can handle it. This one, we're going to watch the video first, then explain, because I found it's a little easier that way. That's why it's at the top of my slide, so I remember to do it first. But we're generally talking about how our bodies absorb nutrients. So. And yeah, rewatching this video, it is on Canvas or through the link in the slides is a great way when you do need to prep for the exam.
Organ function is compromised. In order to carry out normal cell processes, cells employ different nutrients within the body, for example, glucose, amino acids, and lipids. Cells have an ability to sense when nutrients are present, and these signals tell the cell when it is safe to promote the consumption of nutrients. When nutrients are scarce, evolution focuses on conservation, maintenance, and repair, rather than growth and replication. Cells basically monitor the nutrient availability so that they can regulate their activity to balance growth, stress, and the damage that occurs. Nutrient availability becomes deregulated in aging, and nutrient supplies inevitably decline, ultimately affecting cellular function. Our body's natural ability to regenerate tissue and organs.
So what it's basically saying is that there are nutrients in your body. And now we're going to put antioxidants on the side, and we're going to talk about nutrients in your body. And the cells need those nutrients in your body to be able to do a lot of things. So if we come over here to the green circle, there are two main factors that are going to come into this, what I call mTORC1. So mTORC1 is an important, like again, if you were video gaming, you would want it in your backpack. It's a good weapon to have in your backpack with mTOR and mTORC1. So we have nutrients up here, vitamin A, vitamin C, all these glucose, all these different types of nutrients. They're not just any one type. It's all the different type of nutrients. You also have growth factors. So that's coming into like a growth hormone and other things that are other hormones that are pumped through your body. They're not irrelevant, but we're going to kind of focus on the nutrient side of it because we have these come together and that's what affects these mTOR and mTORC1s. And so as we age throughout our life, because this starts at like in utero is when the nutrients matter. So mother's nutrients, your mother's nutrients when you were in utero is what starts this circle here for you, for the nutrient and the growth factors that you had in utero. And so they come from birth up throughout life. And the mTORC-C1, so like dysregulation of the mTORC-C1, signals can either increase or decrease the activation, which like turns on and turns off chronic diseases. So like, in short, if you're gonna get diabetes, it's already in your body. It's there. It's hanging out, having fun. But you're not experiencing it at this point. Your body's still managing just fine for many of you. You don't necessarily have type 2 diabetes that's hanging out in your body. This mTORC-C1 is one of the signaling pathways to help keep the chronic diseases away. They're there, but they're doing nothing. That's why a lot of what we get is genetic, because you're born with kind of the potential for it. But there's a lot that we do that affects whether we turn it on or not, or whether it just kind of hangs in our body, like a book that's never read. Right? You guys probably bought books or DVDs or other things that are just sitting there, video games that are unplayed, that are just hanging. You own them, they're in the house, they're on the shelf, they're hanging out. But these are chronic illnesses. We don't want to take them out and read them, right? We'd like them to just stay on the shelf and hang out there. So the nutrients that are in your body affect the nutrient sensing as well. This mTORC-C1, your cells are looking for nutrients floating around and they're trying to grab it and bring it in. If it floats by but it can't see it, have you ever had like a dirty window? And you can't see very well through a dirty window, or dirty glasses if you wear glasses or sunglasses, like where you just can't see, foggy goggles in the water. That's kind of what goes on at the cellular level, that it can't sense the nutrients. They're there, you could be eating them, but eating the nutrients is irrelevant if it can't grab it and bring it into the cell. So that's what this is kind of talking about. Again, we'll get into more of these in other weeks, but the point of this is that the idea of nutrient sensing is that one of the reasons why our body ages is because it loses that ability to sense the nutrients. And if it can't pull them into the cells can't do what they need to do, which leads to them either senescing or apoptosis, depending on what happens when it doesn't have the nutrients. So the diseases, which also cause problems in the body, metabolic disease, neurodegeneration, inflammatory autoimmune disorders, immunosenescence, genetic or rare diseases, and musculoskeletal diseases. So a lot of people with autoimmune disorders, as we'll learn later on, are also have different nutrients implicated. Often looks in forms of allergies or other things that affect those with autoimmune disorders. Because it plays through this whole path together in the body. It naturally goes in to breaking down. And so it's an aging theory because it's talking about grabbing that nutrients, bringing it into the cell. And while it might not make, it doesn't lead to the body to age, it plays a role in cellular senescence, like it can lead a cell to senesce earlier. But it also can cause diseases that then makes your body have to work harder, which then makes you age faster, right? Like in a way that if you can't manage what's going on. All right, go ahead and explain nutrient sensing at your table.
So. don't-
You can also,
As well as to a, your arms closed, again, as we like, for instance, do that in there. Then or just come to the body, step downstairs. So we, these races.
And open.
So. Yes, which we will put some of this, because it was there.
so. Okay. There's a, putting in property, there's the address. And that's what's considered.
So. Just using that. So. But we said this is what you do. Actually. Years ago. You will know most of that. And I work in town, that's the change. Pretty much. You know what I'm saying? Yeah. So is that explained? Like, is that explained in a better way? It's so easy. Right. So.
All right. Questions about the nutrient sensing?
We need to know the major proponent.
Yeah, I looked that one up. I realized they weren't on there. I will add it right on. So it is, I'll just stick it up here. In a slightly smaller font. This was that. You don't need this other part, but. Because the Whitehead Institute, I mean, there were other researchers working there as well. And this one is from Stanford. We're the ones that did the, oh, you can't see it. You can only see my video. There you go. David Santalini at the Whitehead Institute and Casper Togawa at Stanford. All right. So now, and we have more cellular than we have genetics. So after the break is shorter, especially since I moved one up. So the next one is stem cells and telomerase. And I wanted to keep it here because we talked about telomerase, right? That's that part that can add back to the telomeres that our germ cells have that's healthy. If our somatic cells get it, it turns into cancer. But we're gonna now talk a little bit about the stem cells. So stem cells are the like beginning, the base, which then differentiates into various types of cells. Because they haven't yet decided what they are, they can turn into a lot of different things. We have a lot of variety. And as we age, the number of stem cells we have in our body first begin to diminish, and then they mutate. So even though you have them in older age, they can't do what they could do when they were younger. And that the aging process, like the process of our body aging, kind of mutates and destroys our stem cells that are in our body. We're like, we're older, don't need them anymore. And I'll show you why in the second half a little bit why that actually makes more sense. So as stem cells age, the renewal ability deteriorates and the ability to differentiate changes. It can't. It's not this flexible thing that it was again down here. So internal environmental conditions, things like the DNA damage. We've talked about the mitochondrial dysfunction, right? That was in and epigenetic alterations. All these different things we'll talk about this semester can affect the stem cells so that they do this process over time. I think I'm right at the right spot, right? Go back a little bit.
So that they can regulate their activity to balance growth, stress, and the damage that occurs. Nutrient availability becomes deregulated in aging, and nutrient supplies inevitably decline, ultimately affecting cellular function. Our body's natural ability to regenerate tissue and organs, and repair cell damage, depends on the availability of healthy stem cells. This capacity declines with age, and they become unable to carry out these processes due to exhaustion. Stem cells are also affected by other age-related issues like chronic inflammation and DNA damage over time, which can inhibit their ability to replicate and replace defective cells. This leads to diseases like reduced muscle mass, bone fragility, and immunosenescence, where defective cells are not cleared anymore. As stem cells need to replicate often to replenish damaged tissue, they can't afford to lose their DNA or capacity for infinite cell divisions. They possess an enzyme called telomerase that extends their telomeres, the ends of the DNA, when they get shorter. But the rate of telomerase activity is not enough to compensate for the degree of shortening that takes place throughout life, and thus stem cells eventually senesce or die after they reach their natural replicative limit with age. The mitochondria are organelles within cells that are known to be the powerhouse and the main energy source for.
Okay, we already did the mitochondria. So in the stem cell, it does have telomerase, but is it enough that it does it forever? No, it has just enough to give you just enough telomere back so that it can go a lot longer. Because they're trying to hold those stem cells for as long as possible. Because if you run out of skin cells, you can use it as skin. If you run out of heart cells, you can use it as heart, right? It can be used as whatever cells you need it for. So they're trying to hang on. So they have telomerase so they can go, they don't hit the 50 as fast, right? Like as the telomeres get shorter, before it gets down there, they add a little bit more on, add a little bit more on. So they still will senesce after 50 duplications, but it takes a whole lot longer to get there because they have some telomerase. Even if they get some damage, they've got an ability, they've got better armor on them. Stem cells have more armor, essentially. The telomerase is like an armor, so it can take more hits. But eventually, it will senesce. So the stem cell theory of aging, stem cell and telomerase theory of aging, is the one that says that the reason, one of the explanations for why and how we age is that our stem cells aren't able to keep up and to fix all of the problems anymore.
Is it not such thing as stem cell therapy? Right?
Yeah.
And like, is that like basically like doing what
Your body
that?
could do,
Yeah.
but if it can't do it, so you can take healthy stem cells and then they can turn into any stem.
So like, where do they get those from? From your body or like?
Depends on where. So, there are, here, I'll show you. Here, I'll turn off my phone for ringing too. Okay, it ended. Me. Come out of here. I think it'll come up. I was just looking it up. See if your question does it. If not, I might have to look it up differently during the break. In the textbook. I don't want videos. I know where they come from in the body. I want to see if this gives me the answer I'm looking for. So they're just giving the basic answer. So they can come from yourself or from other people, but they come from the bone marrow most often. But people can donate bone marrow and get it from it. There's research going on. Like that's the stem cell research that is really also because we like the embryonic stem cells. So that's women have in their eggs and other things, you've got unused stem cells to be able to produce. You also have those who've done in vitro fertilization and they have fertilized eggs with even more stem cells that are just sitting in freezers from people who've passed away. And they're just sitting in a freezer somewhere. And the question is, can we use those stem cells to help other people? With the stem cell therapy from, you know, what happens to some of those things. So there is a lot of ethical debates about where is appropriate to get the stem cells from. Can you get them from fertilized eggs that are sitting in freezers that are not going to be used or not? Can you fertilize eggs in a
So like that, like, so like, let's say like you freeze your eggs and, in the future, you still have this like person and like you have some type of disease or whatever. And it can be cured through stem cell therapy, things about like your own egg.
that's the debate.
Oh,
That's also in the debate. So right now the question is, does like where it all comes from. Now, if you can get it from bone marrow from somebody living, obviously you can donate a kidney, you can donate bone marrow, you can donate things. That's where some of it comes from. But some of the best ones comes from younger, because as we can see by our chart, this theory of stem cells and telomerase says that the younger stem cells are better. Remember, they can do more things than the older stem cells can do. So we want younger stem cells, but can children donate stem cells? Like the ethics behind all of it is what you'll see in the news. And in here, I hope to just teach the concept so that when you come with your own opinion, you have a reason to come forward.
So like when you donate, say, kidney or whatever, or you say receive some sort of organ transplant, you have to take immunosuppressants and whatever to make sure your body doesn't reject it. Since stem cells aren't necessarily differentiated into an actual like organ or whatever yet, do you have to worry about your body's rejecting them?
It's not as much. It's like you said, because it's stem cell. So the body doesn't know what to do with it yet. Depending on the state of the stem cell. So embryonic stem cells are the safest to put in a body. And the older it gets, the harder it gets to use. And some of them, if you remember from the very beginning, we talked about there's some cells that don't, like parts of the body that struggle later on. So the main part of this theory is getting at all of that's behind stem cell therapy. So if you hear about stem cell therapy, it's coming from the research in aging. Research of aging actually gives us a lot of medicine. Because we discovered that we have both numbers and their ability and what they do is better when we're younger, and a lot better when we're an infant than even when we become a young adult. So it is a big question about what to use and where we can get these stem cells from. Our own body's doing it by itself when it can. Your body uses its own stem cells to be like, I need you to be this, I need you to be that, I need you to be this. And this theory says that because it has some telomerase, we get them longer, but they're not as long as regular cells. And when they start running out, that's when we start seeing heavy aging. Because we don't have this resource of things to pull from to make whatever we need. We good? I got one more here before we break. And this is loss of proteostasis. And this is the heat shock proteins from those who did the reading. So I'm going to explain it and then we're going to watch it. I know I put it first, but I'm doing it the other way now. Because we've gone through a lot of the things that are here now. So we were actually getting into this with the mitochondria, with the nutrient sensing. So nutrient sensing is talking about general nutrients. Now we're going to talk about, like, you can almost see protein, proteo. We're now talking about proteins in your body. Do proteins cause aging or lack of proteins cause aging? How do proteins in your body affect your aging? So the heat shock proteins play a major role in protecting cells from every kind of stressor. From radiation to infection to oxidation. Like, these proteins are your protectors. So that I like that works together, that proteins protect. That's their job, is to protect. They're in every living organism from bacteria to humans. They promote healthy cell growth and that proliferation, that cell division. It helps it go at a healthy rate and not too fast and not too slow. However, they protect. Protect is not repair. Protect is protect. So it's like a shield can't heal a wounded soldier. It can only protect them from getting hurt. But once they're hurt, the shield doesn't help any. Right? It doesn't heal the wound. They can also help a cell to self-destruct instead of senesce, which can be good later on, right? When we were talking about all those senescing cells building up, proteins can be like, okay, you're done, go into apoptosis. Or a similar thing called necrosis. Both apoptosis and necrosis are ways that it shuts down. But that proteins can help it be like, okay, you're all done. Pop. But if you don't have sufficient proteins, then it just stays in senescent. Like, it goes into senescence rather than pushing it over to an apoptosis. Another role is that it keeps the molecular structure, which homeostasis means balance. So it's called protein homeostasis or proteostasis, right? Like where they put the two together. This is 7. Oh, I left it, didn't I? $7.23.
Failure to dispose of defective mitochondria further lead to cellular damage and eventually aging. Proteins regulate virtually all cellular reactions and processes and provide cell structure. The maintenance of all proteins in their original form, folded in precise, complex conformations, and in abundance, is essential for them to perform their functions optimally. However, with age, proteins are damaged by declining cellular processes. Changes to cellular pH, oxidative processes, or environmental stress can create aberrant protein changes, causing them to misfold and provide inaccurate instructions. They can also form unwanted bonds with other proteins, by aggregating together and thus become toxic. In an ideal situation, aberrant and misfolded proteins are degraded by cellular machinery responsible for recycling, but aggregations make it hard for this to be achieved. These aggregates form clumps that protect the interior proteins from being broken down and recycled. All cells have lysosomes that are tiny sacs of enzymes inside of the cell that engulf and degrade cellular material. Our ability to maintain this process reduces over time and leads to the accumulation of damaged proteins that cause all sorts of diseases like Alzheimer's, Parkinson's, Huntington's, and heart disease. We also have a biological clock in our DNA,
Okay, we did that
which
one
has
already.
an expiry.
So again, the proteins think of it like origami, right? You need it to look like the beautiful origami. But if I folded it, it might be problematic, and that's what happens with aging. It looks like one that I folded instead of my husband. He's much better at that sort of thing. So proteins are folded in your body, and they need to be folded that way. And if they start not folding right, they can stick together, join together. And again, if your body can't keep up with the recycling process and keeping them apart, they build up and cause problems. And so that's
Can also cause cancer.
Can it cause cancer? Well, cancer is the telomere that doesn't come down. So it doesn't cause cancer. But it does, one of them that's really heavy, and as they were saying, is Alzheimer's. Proteins is a heavy, if they build up, they have the plaque build up and the other buildups in the brains, and then you can't signal through the proteins. And then dementias, Parkinson's, other dementias, but dementias in general get hit hard by protein buildup in the brain, and other parts of the body are affected. So it's generally other things. Again, cancer comes more from that duplication happening and not stopping. All right, let's take a break, and then we'll review when we come back and finish. There's just a few more theories to go. Yeah, now is a great time. If you're sitting in here, you can just take a break, but you could also work on your vocab, draw some pictures of things we've talked about today. You could make sure you filled in your theory study guide. And if not, you have questions to ask if you can't get it filled in from the first ones, in the first half. I
Let's do by the end of today.
Which one? Oh, the theory study guides today. But then the vocab is over the weekend. I wasn't sure which one we were talking about. Isn't that Saturday?
Study is all.
Is it all tomorrow? Whatever it says on campus is correct. I have another class right after this one, and I had one before, so I trust Canvas. Whatever it says, yeah, I actually don't like those questions because I have too many things that I put in one day. Yeah.
Missing the major components from the last two theories.
The last two periods, they must be in my notes then. I will put them into the slides. We'll move this down and things.
We would oversee, okay,
Okay.
There'll be a high chance that we will turn most of the stuff tomorrow on Saturday, because I'm still trying to catch up on the one class, and I would like make sure. Okay. On that one, so just want to let you know. Okay, thank you. Yeah.
Ladies.
Sure, that's tuned.
All right, people, let's kind of come back together. I have up here, you don't have to find them in the textbook, for the stem cell and telomerase theory, James Till and Ernest McCulloch. Are the proponents. And then on the proteostasis is Ferruccio di Sosa, Litosa. I'm Italian. In 62. And if you notice, the reason why I put the years is because it plays from that HeLa cells. Remember I said a lot of what we know comes after she died in the 1950s? Lots of things were discovered in the 1960s.
You go back to one.
Yeah, we'll make sure everybody gets this one down and then I'll go back. Previous one is James Till and Ernest McCulloch. And these slides, this version isn't, but I will update and re-put these ones up because those two were missing, but they're in the textbooks as well. But I will update and put it back up. Okay. Well, we're now going to go on to the genetic theories. The only genetic theory we did so far you should have that we did before was the telomere length was technically a genetic theory. So when that comes up in your list for today, it will end up getting skipped because we already did it. So we're now going to talk about DNA. What's going on with our DNA? So this one is one that a lot of you guys will actually think is quite interesting. You didn't read about it. Who's heard about caloric restriction? What is caloric restriction? What have you heard? What counts as caloric? Is it a diet? If you go on a diet, is that caloric restriction? No, definitely not. Next. So what is caloric restriction?
Isn't it like the minimum amount of calories you need to function or something?
In a way, yep, it's the minimum number of calories close while maintaining all of the nutrients you need. All the fat you need, all the protein you need, all the vitamins you need. A diet is reducing something. This one's keeping everything while taking out calories. It's called caloric restriction. And then we got McKay, Crowell, and Maynard in 1935. It often popularizes as a diet, but it's not a diet. So caloric restriction means all of the nutrients that your body needs, but not the calories. So when we have true caloric restrictions, humans don't do it. But when you have true caloric restriction, then what we see is reduced levels of insulin and IGF-1, that's growth hormone, factor 1, that's what helps you grow. Which, remember that mTOR? Remember I talked about mTOR and mTORC1 and the nutrient? So we're going to see it coming up here again. So the absence of glucose affecting the mTOR and autophagy is going to, is part of the process of cleaning up. Remember they talked about in the video all the time how you clean it up, we got to keep it cleaned up. This is the cleanup. That's your recycling process in your body. And all of these, increased levels of all of these, increase living longer. So caloric restriction does not cause people to live longer. But with caloric restriction, reduces levels of insulin, it takes glucose out and enhances the NAD plus concentration, and those in turn affect things that affect longevity. So animals on food restricted diets that still have all the nutrients have lower levels of blood circulating glucose, insulin, and triglycerides. But there's a catch. They're infertile. You can't have babies. Anybody practicing caloric restriction, the true actual definition, cannot have children. It's impossible. It's totally not possible for your body. And it is a near starvation level of calories. We're talking concentration camp level of calories to get this to happen. Anybody ready to sign up?
Oh, no.
No, I'm just kidding.
I'm not gonna lie, this slide, I've had like this slide written down like 4 times already, so I was like, it was just funny to bring it back up. You taught it in,
Yeah.
literally every class I was in. And,
I don't think we did it in family relations,
well, yeah,
but
or 200.
yeah.
Oh, not that one.
Is this like what they do on those shows, like 600 pound, and they take them to the hospital and they say that they're on calorie restriction, but are
Yeah,
they not going by the true term? They're just saying that.
they're probably saying they've restricted their calories rather than that. And maybe some people are doing true levels of calorie restrictions when they're doing it on the show. I know the research where people volunteered to be in and actually do caloric restriction. There were a number of adults. There was a study where it was 2 years, a 2-year study where you agreed, the researcher gave you the vitamins like to get you all of the nutrients you needed. And they did all look like concentration camp numbers. And they ended the study because like it wasn't a lot, even though people consented, they ended the study.
Say like your prime time, guy or girl, right? And you say your age between 25 and 35, right? And then you like are doing caloric restriction, and then so you're saying you're infertile, right? And then say they quit being that, they start eating regular, can they gain that ability back?
Correct.
Okay.
And I'll show that in another one how that works. But yes, it's only in the state. So it delays age-related diseases. It attacks cancer. Your telomerase doesn't function properly, so it can stop the cancer process and other neurodegenerative diseases. It's mediated, which means it goes through gene expression, gene transcription, and the mitochondria. The mitochondria is part of your metabolism, so it affects your metabolism. So we've done it quite a bit in animals, and this is what we find. The best human, this one is for in months. So this one is the mice study. So this is a mice study. Obviously, none of you are planning on living 60 months and being happy with that. This is age in months. Ad libitum means you get what you want. Like they have the food out for an animal and they get to pick when they eat and how much they eat. And when they're full, they can stop. And like there's nobody giving them food, just kind of sits at a table and you come and get it when you're hungry. So that's ad libitum. This is the mean lifespan, and this is the maximum lifespan of eating at ad libitum. If you take and you reduce by 25% the number of calories while maintaining the nutrients, you don't see much change. And this is what most diets do that call themselves caloric restriction. 45% restriction. I want you to think about that. How many calories are you supposed to be eating a day? 2,000 for the average adult man or active woman. So you should know your own. 1,800 for the average adult woman. 2,000 for the average adult man. Half of that. So if we're going with the 2,000, we'll pretend that we'll play to the average adult male. 2,000 is also what everything does. Did you know that the food things are always on the average adult male? Because I don't know, maybe they can't calculate it. I don't know why we don't have 1,800 listed on everything instead. But 2,000 calories, you're down to about 1,200 calories a day if you're doing the 45% restriction. That's like a Big Mac. And that's it all day long.
Sure.
Just one, no fries, no drinks. Big Macs have a lot of calories in them.
You can eat a lot of things,
With what?
2,000 dollars. You can eat a lot of.
Yeah, but if you're down to 1,000, you're out a lot of things.
Half of it.
And 65% restriction is what they generally consider. 80% restriction is what you need to get the actual outcome. So 80% of the calories you need in a day.
2,000.
That's 200 calories a day you're allowed to have on a 2,000 calorie diet.
It's like
So your brain needs like 600 calories to function.
Correct. But you'll live a really long time.
Will I be able to move?
So that's some of the stuff. Is the theory of caloric restriction, does that lead to longevity? The research suggests yes. Again, it's a theory. It's backed up by research. Is it a practical way to live a really long time? Yeah.
Isn't that like the example you gave for like the survivors of the Holocaust, how they live like really long times?
Yeah.
Did this, I'm assuming, but did it definitely have like extra?
Yeah, they do take in exercise calories burned, calories in, calories out when they're doing the research. And as Jonathan was saying, one of the like natural studies we have that's not completely true to form because they were also missing nutrients, were concentration camp survivors. We actually find that those who survived the concentration camps lived on average to be at least 100 years old. A lot of people would have thought being in a concentration camp, and the ones who survived were the ones that were young adults in the concentration camps that lived the longest, because that was a time when their body could handle the, now they still got nutrients. And a lot of times it actually was more like caloric restriction because they didn't totally starve them. They gave them soups that actually had quite a bit of nutrients in them, just no calories. So concentration camps were the closest we have to human experiments and caloric restrictions. They were infertile during those times. There was no menstruation going on in concentration camps. It was impossible. They did not conceive children, even if they'd been allowed to, they couldn't. The bodies, the male's bodies and the female's bodies wouldn't work when you're in that state. And these are, again, still from mice, but looking at the ad libum, again, that's eat what you want. And then the caloric restricted, how we see differences when you do them and things like lymphoma, it was a huge one in the cancers. And of course, nor tumors, which is good, is higher. So it does seem to work. Now, as your professor, I want to make sure nobody goes home and tries caloric restriction. It has a lot of other negative effects on the body.
So it's like basically a high risk, but kind of high reward kind of.
Nope.
No reward, just.
I mean, you'll just live a long time, but you could also live a long time hooked up to an iron lung. Yeah, like if you just live a long time. So I don't know that I would say high reward. However, which everybody likes, is there something called intermittent fasting. Now, a lot of it is also wrong. Be careful what you're watching out there. There's a lot of crazy stuff on intermittent fasting who don't understand what it is, just like caloric restriction. It's related. It is not the same thing, but it does the same thing for a short period of time in your body. It stimulates autophagy. Remember, autophagy is the recycling system in your body. So like it gets rid of, remember how we said it's got to keep up with getting the junk out? And if you keep up with getting the junk out, it's good for your body. Intermittent fasting, so fasting, intermittent, but fasting can stimulate autophagy into your body. So it can make your body start recycling things. It does not mean you will lose any weight. It is not a weight loss program. It is a clean up the bad stuff in your body program so that it slows down the aging process. Because most people who fast do not lose weight. It's not meant to, and you really shouldn't lose weight while fasting. I mean, maybe in those moments you'll weigh less, but over time, because you still need to eat the food that you were going to have. So it breaks down the damaged or unwanted cellular components. Again, autophagy, that's the early stage cellular response to stress. Prolonged calorie restriction with excessive autophagy is harmful and actually puts it into apoptosis and gets rid of the cell that you were trying to make healthy. And that's what also happens in caloric restriction too. It can go too far. So you don't want to fast too much. That is super bad for your body. That's why we call it intermittent fasting. When fasting, remember to drink water. It doesn't have the same effect. If you're getting no drink and no food, you just want no calories. Again, this is not about weight loss. This is about aging. I have to put it there like 100 times and still nobody hears that. They go talk to people about other things. So with it, how many hours do you need to fast? In order for it to work, about 17 to 19 hours. Going up to 24 hours is fine. No benefit from going over 24 hours. You don't like win bonus points for fasting longer. 17 to 19 does it. It's recommended for your body that it can handle without getting into any risk areas once, maybe twice a month. You should not be doing intermittent fasting any more often. Now, there is some catches to this. That's really also fascinating to me, because those of you who know me know I also study religion. Religious groups, many religious groups have forms of fasting worked into their religious beliefs that may work slightly differently, but they still seem to be effective. Ramadan, for example, has a time period where they fast for different amounts of times, but it's a certain number of days in a row. But then they don't the rest of the year, and it still hits the same average we're talking about. In old days, Lent worked this way. Lent doesn't quite have the same thing. Now people give something up that's not the original Lent. But the original Lent was very similar to Ramadan, not quite the same, but it had a lot more similar components where there was fasting involved in Lent. It's actually kind of what the word Lent is, the fast. Other faiths practice times of fasting. Hindus, Buddhists also have times of fasting. Other Christians face those times of fasting. Again, so I find it fascinating as like a religious scholar and a science scholar, biology, that the two actually come together. And those are the people who live a long time. The ones who actually practice that part of their faith live longer than those who don't practice that part of their faith.
So is this like one day of fasting a month or something?
Yeah.
Okay. So what if, because I feel like
And I do this part, by the way. I'll just self-disclose.
personally, like there will be times where I'll eat at night and then like I'll sleep and I won't eat till later in the morning, and I probably hit close to this relatively consistently. Is that damaging, or is that just kind of normal life?
It can be damaging. It depends. It depends on what you're getting. You're probably hitting in a young adult age, your body can keep up with a lot of things. You want to be careful as you age, because then it'll have a harder time keeping up with it. You don't want to be going 19 hours every day without eating. 12 hours or doing it. I mean, you may have times when you sleep that long, right? That's a different thing. I mean, it's still hard on your body. When we get to the sleep part of this class, we'll find that's not the ideal either. But there's differences for sickness and other things too, right? This is a normal processes that we do. And in the morning, all you really need is about 100 calories when you get up. Also, if you're just not a hungry morning person, again, 100 calories, that's like a juice box. Drink a juice box. And take some vitamins because you still need the nutrients. And if you're doing this, I still take my vitamins, or at least a daily vitamin on the day when I fast, or when I'm starting it, so that I still have the nutrients in my body, because you don't want to miss nutrients.
Well, they always say like, why there the saying exists like, breakfast is the most important meal of the day, and like always eat a big breakfast.
We'll learn that moderation is important, so it depends on who you are, how you do it. But you need to have calories throughout the day in your brain and your body to function and all the things you're talking about. It's not just a daily net that matters. And again, the day, if you choose to do this, the day when you fast is hard on your brain. Because you're not getting the nutrients that day necessarily that you need. I mean, I still do my same things, but it's much, much harder on the brain. So if you choose to do this, be careful the days you pick. I generally do it on Sundays because I'm not working. You know, it's a day when I can take the day to go for walks and do other more simpler things. I don't grade on that day when I'm doing it. So that I am keeping that. You want to stay hydrated, because hydration will make a big difference. And that's why we also, you know, people who wait longer to eat in the morning, you really want to stay hydrated. But again, it's not a healthy diet. I know who's saying it and what's saying. I've read all the research. It is not a healthy diet. You can lose weight. It is true. But it'll cause other problems in aging. So if you only want today, you can lose weight today. But if you want it when you're older to be a happy, healthy, yeah, sorry.
No, okay.
Oh, I know that it's, I don't want to say articles, but usually it's explained that the moment you stop, it kind of, like the effects are done. Is that true?
Not when we're talking about repairing the body. Some of it, like it keeps going, but it does, like, because it's in that kind of that starvation kind of state that the body's trying to clean up. Like the state it puts the body in. It's not true starvation because we're not going that long, right? Like that it's kicking it into that. So that you want to be hungry and when you're doing this and feel that, and then it will clean up. But you want to drink water. And some things stay with it, but some of the things stop. So it depends on what part they're talking about. But again, most people doing this are only focused on one aspect. Very few of them are gerontologists that are out there doing it. It's often doctors. And one of the things I have to tell you about medical doctors, they do not have PhDs, most of them. PhD means research. Any other D, doctorate of dentistry, doctorate of education, medical doctor, they're practice doctors. Their job is to do, not to research. Hopefully they can read it, but that's not their job. Medical doctors do not do research. They get people to keep them up to date. They go to conferences to hear it, but they're not researchers. They do like one research project their whole time to learn about it, but they're not researchers. It is not the medical doctors that are the most important to listen to when you're looking at new things like this, because they didn't actually do the research. You want a PhD in something that makes sense, right? A biogerontologist with a PhD in biogerontology. Now that's somebody who knows what they're talking about, even if you don't understand them, right? Like they're the ones who actually can and do read and do research so they can understand, well, it's for these people in this time and not this, and includes that, when what keeps going, what stops. The doctors hear it and then share it. So if you hear it from a doctor, doesn't make it wrong. I'm not saying don't trust your doctor. They're just getting it and then delivering it. And so if you're watching YouTubes of people with MDs, it doesn't mean they know what they might, but they might not, because they're getting it and then interpreting it rather than being able to do it from scratch. Okay, any other questions? The theory of intermittent fasting is that idea that keeping the nutrients, reducing the calories is associated with longevity. feel like I'm missing some slides here. We'll see if they're here. If not, then I will find out where I accidentally put them. Back to our autophagy, right? Cleaning up the waste. Lipofuscin is a dark mixture of lipoproteins and other waste products that accumulates in your aging cells. It's in all cell types. And trash accumulates. Ever watched your kitchen trash fill up? That's what's going on. With time, it fills. And too much of it can interfere with autophagy. Again, if you have too much trash in your kitchen trash, have you ever tried to take it out when it's actually too much and it starts falling all over the place and you can't just close up the bag and it just, where if you'd have closed it up when it just got full, it would have been a whole lot easier. You following? That's what we're talking about here. Too much lipofuscin can interfere with the cleanup process because there's too much of it all over the place. And we see that if you have too much of it, we can measure it in your body. It's kind of a biomarker, which is like a test we can do to see for your metabolic rate, like how often you're able to process things. Because it reflects, it gets in the way of that oxidation and free radicals that we talked about before. So there's too much trash in your body. The antioxidants can't come in and take care of the oxidative issues because there's too much trash. Same, no different in your bedroom where you can't find anything when it gets too messy. Things can't do their jobs. Again, severe caloric restriction does reduce the level. That's one of the ways that it helps. And that's also what we see in the fasting. On days when you fast, it reduces your levels, which means your body can then clean up. That's really what fasting does. Fasting is really associated, that's why I put it between the two, because caloric restriction is an extreme theory. Fasting is not a theory of aging. But I put it in there because people are interested in it. I used to not have it in and people would always ask, so I just added it into the slides. But it kind of falls between the two. In that it's kind of helping because it helps you clean up. I actually want to go, I lost a slide. Okay. I'm good. We're going to cover next time, and that's good. I just thought I might have accidentally deleted it. So I just need. So, epigenetics. This is the theory of epigenetics by Thomas Hunt Morgan. He was an embryologist at Columbia University. He coined the term epigenetics in 1942. So epigenetics is the idea that you have your genes, the genetics in your body, and you have the environment affecting it. And as those two come together, your phenotype, genotype is your gene. Phenotype is the expression, what it looks like when it comes out. And just because you have a genotype, you can have different phenotypes from that same genotype. It's not like because you have this, this is what happens. A lot of people make it sound like, for example, your genotype for your height So most of you know that genetics is a major role in how tall you are, right? How tall your parents are affects how tall you are. That comes from genetics. But you actually have height potential. You don't have an exact height that comes from your parents. Instead, the environment comes in, and the way that that gene is able to express out and act and give you the growth depends on your environment. So you may be taller or shorter than you would have been if you'd have had a different environment. We've seen it often in twin studies. That's how we learned about it, was in twin studies. They're identical twins from third world countries who are adopted. One stays in the third world country, one comes to Europe or United States or Canada, right, comes out to a first world country. And they're identical twins that are can be up to 6 inches different in height. Just because of where they were based. We see that often in immigrants from other countries that are short in their countries. They immigrate to other countries that have more diversity in food and other things that allow for a different environmental impact. And they're taller than their parents and their grandparents, and like they get taller by generation. The most effective place to do that is in Scandinavia. People grow the most height difference happens in Scandinavia. For like immigrants from other short ranged countries. So that's kind of an example of, but it's that idea that the environment affects what's going on. So really how something looks, how your genes express is both from the genes and the environment. No, I was right at it. 10:26, there we go. A little before.
Cell can divide, slowly leading to dwindling populations of functional cells. If you think about it, every cell in our body contains the same set of DNA. So what makes them differentiate into different cell types? Gene expression is modified by the addition of epigenetic markers to the DNA, thus changing the patterns of gene expression in the cell, switching on and off the expression of certain genes in the cell as the situation demands. This is known as the epigenome and can be modified by diet, other lifestyle factors, and pharmaceuticals, but most importantly, the cellular environment. It also changes as you age. As our cells are exposed to more microenvironmental and environmental factors, these chemical modification tags are lost, added inappropriately, or shifted around. And these changes accumulate over time and have been correlated with a decline or alterations in function observed in aging. In aging, as the environment becomes more inflammatory, with various inhibitory molecules released from injured and stressed cells, the feedback loop leads to more and more epigenetic alterations in the genome, ultimately changing the function of the cells. The cells in our body contain all the instructions needed to create proteins that are required.
Okay. So in this one, the way that it affects aging, so that's epigenetics, is that it affects your body. But the way the epigenetics works, as they showed you, is that this happens over time. So if you're women who are much heavier in like an obese sort of a state, will have premenopausal symptoms in their 30s. They're not premenopausal, but they'll start all the way in their 30s because of the weight. And it's the environment affecting the way the gene is expressing itself. Things like exposure to industrial pollutants, sun, eating healthy, all of those things can change how the gene expresses itself. So if you don't have the proper nutrients in your body for your cells, it will affect the nutrient sensing, right? Like it'll affect that ability to do what it needs to do. But it will also affect the DNA's way of expressing other things at the DNA level rather than the cellular level. It also affects the DNA level change it.
So is that like the radium girls? Cause.
Yeah, epigenetics is the radiant girls.
Thank
If
you.
you've seen radiant girls, that would be epigenetics. The exposure of the radiant to the body changed, not just health wise. And one of the things about who's seen the radiant girls? If you're not, there's a movie out there. You should go see the radiant girls. Not exactly happy, but it's the story of young women who worked in labs where they would make glow in the dark watches, so to speak. And so they would paint the radium was what they would use to make it glow in the dark. And they would like put the paintbrush on their lip to make it sharp and then do the dials. And they died of radium poisoning, but we didn't have radium poisoning yet. A lot of people thought radium was healthy. And so Radium Girls tells the story of some of the girls. But one of the things is it was covered up by companies, as were not unexpected in the United States, that we don't even know who those great-grandchildren are. Because one of the things with epigenetics is it goes for 3 to 4 generations. The choices and things that you make affect genetically 3 to 4 generations before it can be washed out. And I have one. I have a syndrome that is associated with alcoholism, and I've actually never tasted alcohol in my life. I come from a very long line of alcoholics. I've decided not to find out if I would become an alcoholic if I started drinking. So I do not drink, but I still have it. My grandfather developed a condition that if you look in the textbooks, it will tell you it only happens with alcoholism, but the gene expression passed on. Because when he had it was young enough that it was before my dad was born, and so my dad got it, passed it on to me, and I passed it on to my daughter. It should stop passing on here soon. But they know that it's because of epigenetics. It was a choice that my grandfather made that affects my health. And that's how epigenetics works, both in our own bodies. But if you think about it, especially as a young adult, it's easy to kind of think about some of the ways it's transmitted. Women are born with all of their eggs. It is what it is, and you don't get more. Every choice and everything that happens to you from conception up into childbirth is affecting whatever child that egg turns into. The sperm in the reproduction section, you make new sperm, but it's all still being affected by the base system. So the same thing is also true for men. So every choice and everything that happens to you will affect any future offspring. And the eggs that I have exactly in me, they're being affected, but also the potential for your descendants is also kind of in there. Because when you're pregnant, for example, not only do you have, for women, the child you're affecting, but the child, if it's a girl or a boy, sperm, whatever's in the sperm potential. But if we think of a girl, that's easier because the eggs are there in a way we can picture. I'm affecting those eggs already. Not just my baby, but my baby daughter's eggs are being affected by the choices I make. So it's always guaranteed to go to the grandchildren. And also the men have the same thing happening. It's just easier to picture with girls. I just don't want the men to think they're off the clear. It still does work. It just it looks differently. It's a more complicated process, but it has the same outcome. So these environmental factors and influence, it includes things like the foods we eat, the exercise, the drugs, the both medications can actually change things. Sometimes people who get on depression medications can actually have a positive effect in their brain before their children are born, and then the children are less likely to get depressed than if the parent didn't manage their depression before they had children. There's a lot of really cool epigenetic things that go both directions where you can have positive effects on the future by the choices that people make. So that's epigenetics. The theory of epigenetics is that all of those things that are coming in, both the genes, but that the environment interacting with it is what kind of plays a role in our aging. But the important thing is the environment gene cross. That's what when you're trying to decide if it's an epigenetic thing, it's gene, any way you can think of environment, not just sunny day environment, but like you can see if they're all influences that are not genetic can come in there. And so we can consider things like the child abuse I was talking about earlier is epigenetic, right? The abuse you experience affects the genes in the telomere length. That's an epigenetic studies that look at that. All right, why don't you go ahead and describe, we've got a couple of them here. We won't do that. We'll do I'll just do epigenetics for now, and then I'll do the next ones for the other ones. So go ahead and describe epigenetics at your table.
When basically genetics
Just right there.
and
So that
environment together, like
high rate.
those
Related to that. And then genetics, that's all. Parents play a part in that. And you're made how.
You have like a
Basically.
but say you live
On the
in
easier way.
a specific area. So that area is actually shaped physical art.
Any questions for epigenetics?
Does it only affect phenotype or does it also genotype?
It only affects phenotype. The genotype comes from the genes. Because you can inherit phenotypes as well. You can inherit both genotypes and phenotypes. And that's where the confusion often comes in, because you think you only inherit the genotype, so it must affect the gene, but it doesn't actually affect the gene. The gene is still the same. It's the expression of the gene can be passed on. And that's what we didn't used to know. We used to think only genes got passed on. We didn't know gene expression could be passed between generations. All right. My last theory for today is genomic instability, the theory of genomic instability. And this is kind of like, I see it as more like the happy side of it, because we've been talking about everything falling apart in all of these theories, because that's we're trying to predict aging. And this is a theory that deals with repair, of how it's the theory that explains, because it explains phenomenon. The phenomenon is how DNA repairs itself. And so we have some primary ones, including base excision repair, nucleotide repairs, mismatch repairs. I'm going to show you the video, but I wanted you kind of to pay attention to these different types of repair. In the DNA, again, this is not cells. This is DNA level repair. Down at the DNA level.
Changing the function of the cells. The cells in our body contain all the instructions needed to create proteins that are required to maintain the body structure and function. However, the genome is under constant attack from both external sources and internal environmental factors. Fortunately, DNA also encodes a number of processes that detect and repair virtually all of this damage. But repair is not perfect, and as we age, DNA repair mechanisms become less effective. So more and more damage from imperfect repairs to our genome accumulates, and the effects of these mutations compound. This changes the function of the cell, and these changes are transferred into each future copy of the cell. Cancer is one result of unrepaired DNA damage or incorrect repairs. While mutations can occur at any point in time, they are probabilistic events, so the longer you live, the more likely that this is to happen. Genomic instability is arguably the biggest driver of aging. Ultimately, it affects the ability of our body to produce essential functional proteins that are needed to carry out various functions. Whether it's facilitating biochemical reactions, maintaining the scaffolding, keeping your cells together, or the cell-to-cell communication, proteins are involved. So accumulation of DNA damage with age affects all of these natural processes. There are clinical trials testing treatments for all of these hallmarks at various stages of development. Only time will...
Okay, so with it, this one is last today because it kind of is an implicator in all the other ones too. Because if the DNA doesn't replicate, remember we talked about it replicating, if it doesn't do it correctly, we have DNA damage. We have repair systems that will fix it, but those aren't as good over time. And so then when you get a wrong thing, it sends the wrong stuff, and then it starts not making the proteins right. Instead, it's doing something else, and then it's not autophagy isn't happening properly. And this is because the DNA is not getting the right thing, not the information, but it's not even correct. The DNA is not correct anymore. So that's one other way. When I said there was another way we were going to get to cancer, is that it can be, and one of the things that can happen is that the DNA can accidentally forget to say don't use telomerase when the DNA is damaged. So it can be damaged in childhood, and that's where we often get childhood cancer. But it's more likely to be damaged as we age, and so there's a better shot that the telomerase doesn't get told, because you have all of it. Remember, your cells, your DNA is a copy of everything. All the DNA is all the same. It's just telling it what to do with each cell. So a somatic cell, which isn't supposed to have telomerase kicked on, the DNA is like, oh yeah, just turn it on. And then instead of something coming in and saying, no, don't do that, that was wrong. As we age, there's a higher likelihood that that gets through. And that damage continues, and then it can cause cancer or other things. However, if an error burden is too severe, the cell will also go through apoptosis. So that can happen, but sometimes it can do a lot of damage before it gets to that point. Right, like getting from damage to severe can send a lot of wrong messages in between. Any questions? All right. Work to get these ones in next week. Theories are a little lighter. I started with some of the harder ones so that we could also review them as we need to a little bit next week. But make sure you get them all in there. These are the notes you'll have for the exam. Because my next one says end of class, right? It made me stop, even if I wanted to keep going.
Do you need us to submit anything for the in-class assignment?
No, I took the pictures. So I'll be good.
I can ask you, so that's like notes of the lecture. I didn't do the core notes.
Yep, that's okay.
Can I throw these at the end of a study?
Yeah.
And submit that? Okay, fair.
Yep, that's fine.
Just for my own, like, curiosity, when you do the intermittent fasting,
Yeah.
do you generally like start from the time you stop eating, like say the night before and sleep, and then just tack on, or do you?
No, I usually skip dinner
Okay.
and then go to dinner the next day. So I eat dinner,
And then,
like I eat lunch,
right,
and then I'll eat dinner the next day.
okay, that makes sense. So I was trying to figure out how, like timing wise, how that would work.
So that I'm missing 2 full meals.
Okay,
Like I usually miss dinner, or 3 meals, like
Yeah.
dinner, breakfast, lunch,
right,
and then I eat dinner.
okay.
That's how I do it. And I don't know that's the right way to do it, but that's the way that I have found works best for my body. Because then the idea is that I still want to make sure I'm getting the nutrients every day, and that helps me make sure I am eating
Yeah.
with like, because then you may be going longer, right? Like the way that it's working out. And
That makes sense. Thank you.
Let's do. The what?
like the theories that we did.
Or, yeah, tomorrow, Friday, yeah. So, yeah, tomorrow at 12 is my interview, so.
Okay.
We'll see, and Professor Lorenz just sent me like a whole bunch of things that I should